Hi, my name is Henry Kavanaugh, and I'm a junior chemistry major at the College of the Holy Cross. This presentation summarizes my experience doing research in Professor Linton's lab during the Weiss Summer Research Program. My summer was split between two primary projects. The first, to work through the synthesis of a beta sheet mimic molecule, and the second, to learn techniques that allow me to quantify subtle differences in hydrogen bonding. These techniques all make heavy use of the college's NMR, and I gained a lot of valuable familiarity with the instrument. However, before I get too deep into these studies, I think it makes the most sense to discuss the beta sheet mimic molecule itself. This molecule was designed and has been shown by our lab to mimic the internal hydrogen bonding patterns of larger beta sheet structures. The utility of the mimic is rooted in the fact that proteins and polypeptides containing beta sheet structures are extremely prone to aggregation. While this aggregation is a large part of scientific interest in beta sheet structures due to correlations with diseases such as Alzheimer's, it is undeniably a frustrating challenge for researchers to grapple with. Aggregation causes molecules that may not have been particularly soluble to begin with to come out of solution, and this interferes with many different potential methods of study. However, our small tripeptide dimer is very intentionally designed to overcome these problems and to dissolve and stay dissolved in organic solvents, enabling a multitude of useful and sensitive experiments. One important aspect of the molecular design is the hexanyl tail that has been attached to one of the ends of the tripeptide. This tail greatly increases organic solubility and stands in place of what could have been a potential hydroxy hydrogen bond donating group, one which could have contributed to aggregation. In a similar thought process, the other end of the amino acid, at what could be called the N-terminus, the nitrogen has been methylated, removing two other potential hydrogen bond donors. In the center of the molecule, a disulfide tether holds the monomers together. As I will detail a little bit later, this tether was formed by dimerization at a central cysteine residue. Because of the small size of the molecule, it is necessary to have this component so that the molecule does not simply fall apart. Finally, at least in terms of the design of the mimic, there are four variable R groups that can be manipulated for the purposes of study. Most commonly, these groups are matched on either side of the dimer, and the first spot is filled with leucine, which can help to enhance organic solubility. Our group has also been talking about constructing asymmetrical mimics, but we have not tried that quite yet. As far as the other R groups, I chose to look into the potential impacts of threonine and serine, specifically with interest in their potential to act as hydrogen bond acceptors to a nearby amide. The benzyl variations were chosen to prevent activity as hydrogen bond donors. I don't have data that I think is meaningful enough to share yet at this point, but I believe that I will very soon. So I just want to really briefly touch upon the primary mechanism of the synthesis. Most of the steps are extremely similar and make use of the coupling reagent EDC and the nucleophilic additive oxima. These are used in conjunction to activate carboxylic acids, allowing for amine nucleophiles to react and form a peptide bond. This is the general mechanism up to the point where it becomes clear why we need the additive reagent. At this point, there are two potential pathways. The one without oxima involves an intramolecular reaction that creates a useless urea, while the path with oxima eventually results in the desired bond. This mechanism is repeated throughout the synthesis until we get to the dimerization step. This step instead utilizes a simple double substitution mechanism, where one of the cysteine residues attacks the iodine reagent before being attacked by the other residue, with the iodine now acting as a leaving group. So now we have finally gotten to the hydrogen bond quantification studies that our lab uses. First up, are concentration studies, which are done by serial dilutions of a solution that contains the molecule of interest. At each dilution point, an NMR is taken so that we can observe the downfield movement of the hydrogen that we are interested in. The resistance of a molecule to this shift suggests a relative strength of intramolecular hydrogen bonding, as hydrogens that are more sensitive to concentration changes are taken to be more prone to intermolecular interactions. The graph before you is all of the concentration data that I collected this summer. And while the exact molecules aren't quite relevant to this presentation, they do give clear examples of sensitive and insensitive results. The second type of study that we perform is concerned with solvent interaction. Solutions are prepared in C6D6 and gradually increased in their percentage of DMSO, which is a more polar solvent that is capable of accepting hydrogen bonds. Just as before, NMR scans are obtained after each addition. These studies operate under very similar thinking to the concentration studies in that molecules that are more sensitive to the introduction of the polar solvents are taken to be experiencing weaker intramolecular hydrogen bonding. The next two studies were not as central to my summer as concentration and solvent studies were, but they are definitely worth mentioning. Hydrogen deuterium exchange is a common study that is done across the world, and it investigates the rate at which hydrogens exchange for their deuterium isotope. This again is taken to be proportional to the level at which the hydrogen bonds are tied up in intramolecular hydrogen bond interactions. The data on the screen is from our lab, but it is not mine. It's taken from an old paper because we didn't really focus on hydrogen deuterium exchange over the summer due to the promise of a new NMR in the fall. Finally, reaction kinetics, in the context of our lab, refers to a study that I was trying to develop at the end of the summer in order to investigate the effect of hydrogen bonding on carbonyl hydrogen bond acceptors. The graph on the screen is my initial result when trying to set up this type of study, and I hope to build off of it this fall and get some really interesting results from this type of study. The data itself is collected by comparing the relative size of specific product and reactant NMR peaks as they change over time. And that is the end of my presentation. I wanted to say a special thank you to Professor Linton and also Janice and William F. McCall Jr., whose generosity has allowed me to have this amazing opportunity to learn about incredible things such as the NMR instrument, flash chromatography, and peptide synthesis. It really would have been impossible without their generosity.